Good afternoon for Seoul and good morning for Europe. It's a pleasure to be here, and I'd like to thank the organizers of this conference for the invitation to participate in the meeting that they are discussing, where they are discussing very interesting topics regarding food and antimicrobial resistance. The topic of today, we have to highlight that we are here because we are facing an antimicrobial resistance healthcare crisis. And in fact, antimicrobial resistance remains a major public health concern in the European region. And this is showing here with the data that we have of 670,000 infections due to resistant bacteria per year, and approximately 33,000 people dying as a direct consequence. In the United States, a similar number of deaths per year have been also associated with infections by antimicrobial resistant bacteria. And also more recent data from the United States shows a remarkable increase, at least of 15% in 2020 in the hospitals. And this is associated, this number, and this increase is in the number of infections that are causing, the number of infections associated with antimicrobial resistance and the number of deaths. Regarding data from worldwide perspective, this global estimates from 2019 points to 1.27 million deaths directly resulting of AMR, meaning right now that drug-resistant infections kill more people than HIV or malaria. And a 2016 review on antimicrobial resistance estimates that by 2050, as many as 10 million people could die each year as a result of antimicrobial resistance. As a consequence of high amounts of antimicrobials that are used in animal production and human health care, these two settings are the main hotspots for antimicrobial resistance selection and spread. The spread of antibiotic-resistant bacteria could occur, as you know, by direct contact between and within animal and humans, but also through food or the environment. And although the environment importance is increasingly recognized, the food-producing environment is not so well, and its contribution to the antimicrobial resistance burden is insufficiently characterized. This need for further scientific knowledge on the contribution of the food-producing environment on antimicrobial resistance, emergency and spread, as well as on the impact and of strategies and options to mitigate antimicrobial resistance, was also recognized by the European Commission. Therefore, the European Food Safety Authority undertook a self-task mandate with the collaboration of other European agencies, namely the ECDC, the European Medicines Agency, and the European Environment Agency, together with external experts, to advance on the understanding of the role and the mitigations that could be applied to food environment setting. A summary of the main findings of the EFSA scientific opinion on the role of the food-producing environment in antimicrobial resistance, the emergency and development are included in this presentation. So firstly, I will present the main risk factors for occurrence and persistence of antimicrobial resistant bacteria, following the main sources and transmission routes leading to the contamination of foods of animal and non-animal origin with antimicrobial resistant bacteria and or the resistance determinants, and then the strategies and options to mitigate the risk of emergence, spread, and foodborne transmission of those antibiotic resistance bacteria, resistant bacteria and antibiotic resistance genes will be also performed, presenting some evidence of successful measures. Following data gaps in these topics and future research activities in this area will be also discussed. And in this presentation, the food sectors that are considered and that all these aspects will be discussed are the plant-based food production, including fruits, vegetables, and other crops, as well as the terrestrial animal production, including in this case poultry, cattle, and pigs, and aquaculture. General factors and more specific ones, more specific bacterial traits, contribute to the occurrence and persistence of antibiotic-resistant bacteria or gene. It is well recognized that antimicrobial selective pressures is a key factor for occurrence and persistency of these threats, either by, in the case of antibiotic-resistant bacteria, either by promoting the emergence of resistance by selecting spontaneous resistant mutants, or by promoting the persistence of resistant bacteria in detriment of susceptible ones. We know also that the inadequate definition or implementation of security measures are also important on the spread and persistence of these threats, and in which concerns specific features of the bacteria as biofilm production or the ability to also tolerate biocides, metals that can be used as feed additives. These are traits that confer an advantage to those bacteria, and that leads also to the persistence on those environments. So these, together with the replacement of animals and persistent environmental contamination that is associated with this ability of the replacement of animals in place where there is a persistent environmental contamination, contributes to the continuous cycling of bacteria between animals and their environments. We have also to highlight here as an important factor for this persistence of antibiotic-resistant bacteria, and even for the occurrence of novel genes in bacteria of relevancy, the contribution of the natural microbiota, as already been described, that several genes, resistance genes, originate from bacteria from soil and water bacteria. And one of the examples is the CTXM genes that confer resistance to very important antibiotics, the third generation cephalosporins. From the representation of the main AMR sources and transmission routes that were identified in the food producing environment of the three food sectors that I mentioned before, and also here the human compartment that also was assessed in order to connect or possibly assess the connection with the food producing sectors. So from this representation that we have here in this figure, where the team involved in the scientific opinion with the data available on the literature draft, this representation is really possible to show that food producing sectors are linked with human sources and animal and environmental sources of antimicrobial, where antimicrobial resistance is really shared and in a cyclical manner. one of also an overall conclusion about these sources and transmission routes that affects all these settings is the fact that all food producing environment sectors and sources they contain antibiotic resistant bacteria and antibiotic resistance genes. So this means that for each sample type from each source, of course, there is already description in the literature of the antibiotic resistant bacteria and antibiotic resistance genes. And here we are contextualizing this assessment on the European area. This is also an important aspect that I should highlight as the opinion was focused on this region. So also one important overall conclusion from this appreciation of the data regarding sources and transmission routes of these three sectors is the fact that antibiotic-resistant bacteria and antibiotic resistance genes, as well as substances with antimicrobial activity, are indeed introduced to animal and plant-based food production environment, mostly through fecal waste, and fecal waste that could be of human or animal origin. Also, irrigation and surface waters were identified as transmission routes of fecal antibiotic-resistant bacteria and antibiotic resistance genes of animal and human origin. And in this overall first appreciation of the data, it should point out that also a large number of data gaps exist in the relations to the sources and transmission routes of these antimicrobial threats in food-producing environment. And these include diversity of antibiotic-resistant bacteria and antibiotic-resistance genes, as well as mobile genetic elements that could be associated with the dissemination of those threats. In fact, we lack the demonstration of the introduction of antibiotic-resistant bacteria and antibiotic-resistance genes from most environmental sources into the production chain. So this is another important point. Is not we might have description in some sources of antibiotic-resistant bacteria or antibiotic-resistant genes, but the connection, the linkage between those description and the food that is consumed is often absent. So this is also an overall year first conclusion from the data that was available. And also the quantitative impact of contamination of the European production environment on foods or public health is also not known. Considering now in more detail the sources and routes associated with the plant-based food, apart from what was already mentioned, that major source and transmission routes is fertilization and irrigation, where fecal origin, manure, irrigation, and surface water are here the most common contaminants. Also, it should be here highlighted that in this scheme, the potential sources and transmission routes that were identified for this sector were here represented, and those include the soil, the dust, the farm animals, the wildlife, arthropods, workers, equipment, and processed water. And this adds to the major ones that already I talked about. And it should be also noticed here that the load and type of antibiotic-resistant bacteria or antibiotic-resistant genes contamination is influenced also by crop characteristics and the agricultural practice. As for instance, the use of reclaimed water could influence what is the level of contamination of these plants growing on these soils. A particular focus of the EFSA opinion was on the assessment of the presence of highly priority for public health antibiotic-resistant bacteria and antibiotic resistance genes. And this meaning that with this designation, meaning pathogenic bacteria resistant to last resort antibiotics and commensal or environmental bacteria with mobile genetic elements conferring resistance to last resort antibiotics. Those examples include the ones that are here represented. Namely, you can see here the vancomycin-resistant fecalis and also the extended spectrum Escherichia coli, among others, and all with a particular focus on those highly priority antibiotic-resistant bacteria and antibiotic resistance genes. The literature was searched for looking for description of this type of traits. And you can see that in the plant-based food sector, there is already some descriptions of sources contaminated with this type of bacteria, but there are really few studies looking for antimicrobial resistance in different sources of plant-based food, and also these effects, of course, also the description of these high-risk bacteria, as they are also quite few, the descriptions available. In which concern the terrestrial animal sector, and in addition to animal fecal waste that was already previously mentioned as an important source, also feed the farm workers, air dust, rodents, equipment, and visitors were also identified as sources. Other potential sources identified and considered of higher relevancy for animals kept outdoor as compared to the ones on closed facilities include pastures, soil, surface water, drinking water, air, dust, wildlife, or other domestic animals. Sorry. Skip to the, okay. And in this case, highly priority antibiotic-resistant bacteria or antibiotic resistance genes have been reported in different environmental sources, although limitations due to the methodology strategy does not allow us to have a complete also picture of the antibiotic-resistant loads and types. So, although this is terrestrial animals is one of the sectors where we have more descriptions, they are much more associated with the animal or even with food, less with the food producing environment. And in any case, the descriptions available for food producing environment, they have still limitations, and most of them, several of them at least, are associated with the methodology strategy that was used in the study. In which concern aquaculture, full information is available for this sector, and for aquaculture, water, sediments, and feed were identified as sources. Wildlife workers, ice, and equipment were considered potential sources, and the main transmission route identified is water. Here is observed previously for the plant food sector. We have also a paucity of studies, And this affects also the description of highly priority antibiotic resistant bacteria in the resistance genes, just with a few examples regarding fluoroquinolone resistant Enterobacterialis and extended spectrum cephalosporin in E. coli, resistant E. coli. And this is, as I mentioned previously, in the terrestrial animals, and you can just show again, in the terrestrial animals, although not complete, the picture is not complete, we have much more description of these important traits in different sources of those food sectors, and here you have a more extensive list of bacteria belonging to these critical ones, priority ones that were found in the studies. In any case, as I mentioned, there are problems in regarding comprehensive analysis of types and also loads of different bacteria in this setting. So right now we move to the mitigation and strategy options that are available and possible impact of those mitigation and strategy options. And it was identified and is also shared already by a large community that prudent antimicrobial use is the most important measure to mitigate antimicrobial resistance that is applicable for all food producing sectors, and together with correct implementation of effective general measures that includes good hygiene practices and biosecurity, this could prevent and reduce the occurrence and transmission of pathogens and other microorganisms. Also, when available and with proved efficacy, the use of vaccines or probiotics, as they can reduce or prevent the infections. So therefore, this is highly recommended when available. Also, biological methodologies has been the focus of the several research studies that target also the mitigation of this type of problems. But they are still in a very early phase of the development, and right now is not a solution for practical purpose. Activities at production stage, which can widely disseminate large numbers of antibiotic resistant and resistance genes in the different production sectors, are really a priority for intervention. And for all sectors, reducing the likelihood of introduction, dissemination, persistency of fecal contamination, this should be really a priority. More specifically, for the plant production, reducing the bacterial content of manure, sewage sludge, and irrigation water is important. And for livestock production, preventing transmission from other animals, either rodents, arthropods, or wild birds. Dust also is important to prevent transmission from dust, from feed, or surface runoff of water, as well as proper implementation of cleaning and disinfection and hygienic procedures for workers are considered relevant. In the case of aquaculture, ensuring high microbial water quality by reducing or eliminate antibiotic-resistant bacteria and or antibiotic resistance genes in wastewater effluents, and measures to prevent feed contamination are also considered priorities. One of the case and the examples of the success of a mitigation measure, this is the case, and we have several evidences of this success for the reduction of colistin in animal production. Colistin is a last resort antibiotics for human infections due to several gram-negative bacteria. And the use of this antibiotic in animals as well in humans can select for resistant bacteria. Especially of concern in this case is the selection of colistin-resistant gram-negative bacilli with transferable resistance genes conferring resistant to this antibiotic. These are called the MCR genes. And one of the successful examples of these mitigation measures was reported from Spain. Colistin in this country has been commonly used in the swine industry to treat diarrhea in piglets, and following the European Medicine Agency's advice about the use of colistin in animals, in 2016, a strategic and voluntary ban of colistin was sorry, because it's too sensitive right now. I hear the mouse. In 2016, a voluntary plan to reduce the colistin consumption in pig production was launched in this country. This initiative was followed by the 80% of Spanish pig producers, contributing to a decrease in colistin consumption of 97%. And in this, in a recent study, where this graphic came from Miguel Iveloto, they evaluated the trend of colistin resistance mediated through the MCR1 mobile gene, here represented. I will include here. So here represented in bars, this MCR detected by PCR are here represented in bars. And they evaluated the trend of colistin resistance mediated through this gene, this mobile gene, in Spanish food producing pig population, and compared it with published polymyxin sales data in veterinary medicine. And these are the lines that are here in yellow. And they wanted to assess their possible relationships. The data demonstrate a connection between polymyxin use and colistin resistance, with a decreasing trend of colistin resistance. Here you can see this. So this trend of colistin resistance is also connected with decreased use of colistin after a period that before this ban, it was observed an increase in the consumption. So the data demonstrate a connection between polymyxin use and colistin resistance and an efficacy of the ban of colistin in this period. We, with a decrease, sorry, because it's not running right now. Let me see what happens right now, because I have more examples. Maybe I have to turn off the laser. Okay. Oh, it's running. Okay. So other examples of reduction on MCR occurrence was described after extended periods of colistin ban, and this was described in Portugal in pig farms. We observed a decrease. We observed, sorry, no. We observed a decrease. We observed a decrease from 98% of positive samples with MCR in 2016 to 27.5 in 2018. Also in Great Britain, reduction of MCR positive also pig farms was observed in 20 months, and in China for pigs and chicken, a reduction to a level lower than 50% was observed 2 years after the ban of colistin in 2017. There are less reports on this effect of the colistin ban, and after a short period of after this the ban. But this is a paper that is from a group published in 2021, where we describe in poultry meat the reduction from 60% to 24% in 3 and 8 months, respectively, after voluntary colistin complete withdrawal. And in same farms, poultry meat from same farms, 1 year later, there is no detection of MCR1 in those poultry meat from the farms and the study. Another example of the not only of the mitigation strategies, but also the need that we still have for certain sectors of having first the surveillance study, is the example associated with the plant sector. And this plant sector is, as already mentioned, a sector where the risk of contamination with antibiotic-resistant bacteria and or resistance genes starts at pre-harvest stages. And as also mentioned already before, there is a lack of data on crop contamination sources and transmission routes to guide effective strategies of the mitigations. So in Portugal, in 7 greenhouses producing lettuce in the north of Portugal, we conducted a study to detect antibiotic-resistant bacteria, CTXM and MCR gene in lettuce greenhouses environment. Sampling comprised lettuce soil, all treated with manure, except one, and 5 to 6 months before first sampling period, this soil were collected, and also we collected water from the same food-producing environments. The data that we obtained points to the soil as a source of resistant bacteria, with higher resistance rates observed for tetracycline, sulfonamides, ciprofloxacin, and the penicillin. The highly priority antibiotic resistant, and this, I think that you can see here that the bars associated with soil are the ones that are here in brown color, and this really evident, this higher contamination with antimicrobial resistant bacteria for soil comparing to the other samples that were taken from these greenhouses. So as I was mentioning, the data obtained real points for the soil as a source of resistant bacteria, not only to antibiotics like tetracycline, sulfonamides, ciprofloxacin, and the penicillin, the ones where we saw the higher rates of resistance, but also those samples presented resistant to third generation cephalosporins, here represented by CAR and CTX, and also these samples and these isolates that present this resistance to cefotaxime and the colistin, here represented by COL, they contain the genes, the mobile genetic genes, MCR and CTXM. And not only soil was contaminated with these bacteria, but also lettuce was also contaminated with an MCR producing E. coli. And the particular aspect also related with this soil that is worth to mention is that they were soil treated with cattle manure that presented these MCR and CTXM producers. Although several samples, water sample, presented unsatisfactory quality, microbiological quality, mostly comprised non-resistant isolate. So this study suggested that mitigation interventions in this case should target soil and manure. Despite a large number of studies that have investigated the occurrence of antibiotic-resistant bacteria and antibiotic resistance genes in livestock and food, especially in livestock and food from producing animals, the role played by the environment is not sufficiently researched. So priorities include the need for One Health-based integrated studies, long-term longitudinal cohort studies on highest priority antibiotic-resistant bacteria and antibiotic resistance genes. Also, in terms of research priorities, is urgent to validate the efficacy of practical mitigation methods. This is one of the aspects that also was highlighted from those committee involved in this opinion, in-depth opinion. And this include the animal feed and fecal waste treatment. Also, these studies is suggested that they should be linked to the assessment of the effect of the policy developments in Europe that are developed in Europe, namely within the EU Green Deal and veterinary medicines products regulation, that this type of developments, policy developments, are affecting the food producing environments, the antimicrobial resistance, the antimicrobial use, and also climate change impacts. So these studies really should be all the studies assessing the contamination in detail of food producing environments should consider these future policy developments in order to also consider the interplay of different policies in the contamination of the food producing environment. And as part of this Green Deal roadmap, the EU Commission has launched this farm to fork strategy. And among these 4 objectives that are comprised on this Green Deal for Farm and Fork is included, as you can see here, is the 3rd one, is included the reduction of antimicrobials in 15% by 2030. So it is expected that this will positively impact on the MR in the environment also, and contributing to the tackle of the antimicrobial, to the restriction of antimicrobial resistant development and spread. As a take-home message, I would like to mention that design of appropriate strategies to tackle antibiotic resistance requires knowledge on dimension of the problem, and is very important to quantify, to have, to cover the diversity of antibiotic resistant bacteria and antibiotic resistance genes that could be involved here in this problem. Also to know what is the real impact in this case of food producing environment in public health. And then when you consider priority for intervention, biosecurity, infection prevention measures, and when available, these should be prioritized. Also, antimicrobial use reduction in humans and animals should be also a priority, and measures that could impact on the efficacy of the reducing fecal contamination, namely wastewater treatments, are also priority measures for intervention. Sustainable new productions will lead that are connected to low use of antimicrobial and also antimicrobial resistant bacteria should be also promoted. And there is also the need of studies in specific sectors, farms, or in geographic regions to identify main sources of antibiotic-resistant bacteria and the presence of highly priority resistant genes together with One Health integrated studies that could really help on identification of the key sources, key points for intervention. So with this point, I thank you. I'd like to thank you this opportunity for participating in this event and letting you with this beautiful picture from Porto City in Portugal. So thank you.